Hey everyone, it's protein time. Professor Ben here, and we're going to dive into the fascinating world of protein classification. So, my empirical study shows that by understanding their classification, it will help us grasp their diverse functions and roles in biological systems. For today's discussion, we hope to define the following learning outcomes. So, at the end of the lesson, the students are able to first differentiate the classifications of proteins according to chemical differences, activities, shapes, biological importance. Second, match the different proteins to their respective classifications. And third, relate the classifications of proteins to real life situations in medical technology. So let's delve more into the world of proteins. When we think of proteins, we often think of it as an essential component in the human body, serving diverse functions and other biological processes. But did you know? Proteins can be classified into several categories based on their activities, chemical differences, shapes, and biological importance. This classification system not only organizes our knowledge but also helps us to predict the behavior of proteins in different conditions. First up, let's talk about the classifications of proteins based on their activities. Proteins can be classified as either passive or active. So, passive proteins are those proteins which do not have the ability to bind with other molecules as they do not require energy input to carry out their functions. An example of this is keratin and albumin. They do not require or have the ability to bind with other molecules nor require energy input in order to perform their specific tasks. On the other hand, active proteins have the ability to bind with other molecules and requires energy to perform their specific functions. An example of this is hemoglobin. Hemoglobins have the ability to bind with other molecules such as oxygen, carbon dioxide, and other groups such as sulfides. Aside from that, it also requires energy in order to perform their specific functions. So, in order for you to understand more about the classifications of proteins according to activities, I'll be showing you guys a video.
Water isn't a very large molecule, but its polar structure makes it unable to slip through the membrane on its own. In facilitated diffusion, the cell uses channel proteins to allow large and/or polar molecules to pass through. Water diffuses through the cell membrane using special channel proteins called aquaporins. Whenever water diffuses across a membrane, we call it osmosis. To continue with our chart, in facilitated diffusion, particles still flow from high to low concentration. They do use protein channels, but they still don't use ATP. Next, we're going to move into the realm of active transport. Active transport requires the input of energy, usually in the form of ATP. Remember that ATP, or adenosine triphosphate, is a rechargeable, renewable source of cellular energy, almost like a battery. It stores energy in its bonds, and that energy is released when the bonds are broken. The ATP is reduced to lower energy ADP.
Alright, I hope you've learned something from the video. Before moving on to the next classification, let us first have a pit stop. So flash onto the screen are three different kinds of proteins. This was already given as an example a while ago. On the left, we have here a hemoglobin, on the middle is keratin, and on the right is albumin. So I'll be giving you guys 10 seconds to guess their classification according to activity, or you can pause the video for a longer time. So, time's up. Here are the answers. Hemoglobin is classified as an active protein, while on the other hand, both keratin and albumin are classified as passive proteins. So, if you guessed that right, please give yourself 3 big claps. But if you ever did not, still give yourself a clap for trying. You could always get it right next time since learning is not a race. Now, let's move on to the classifications of proteins based on their chemical differences. So proteins, according to their chemical differences, can be classified as either simple proteins, conjugated proteins, or derived proteins. So let's talk about them one by one. So first, we have simple proteins. These are proteins that are basically composed of amino acids joined together by peptide bonds. They are further classified based on their solubility in different solvents and as well as their heat coagulability. So for example, we have here albumin. They are readily soluble in water, dilute acids, and alkalis, and are also coagulated by heat. One fun fact is that they are actually deficient in glycine. Another example we have is globulin, in which they are insoluble or sparingly soluble in water, but their solubility greatly increased by the addition of neutral salts like sodium chloride. We also have here prolamins, in which they are insoluble in water but soluble in 70 to 80% aqueous alcohol, and upon hydrolysis, they yield much proline and amide nitrogen. There are also glutelins, in which are insoluble in water and absolute alcohol but soluble in dilute alkalis and acids. Aside from that, they are also called as plant proteins. To add on, histones are soluble in water but insoluble in ammonium hydroxide. So these are small and stable basic proteins and contain a fairly large amount of basic amino acid, which is histidine. Let us also not forget protamins. These are the simplest proteins and are soluble in water but are not coagulated by heat. They are also basic in nature due to the presence of arginine. Lastly, albuminoids. These are characterized by great stability and insolubility in water and salt solutions. They are called albuminoids because they are similar to albumin and globulins. The second classification of proteins based on chemical differences, we have here conjugated proteins. So under this are simple proteins that are linked to non-protein groups or also known as prosthetic groups. Hence, when hydrolyzed, these produce amino acids plus a non-protein groups. So here we have examples. First, we have chromoproteins, which are pigments, mucoprotein, which is a carbohydrate, a glycoprotein, which is also a carbohydrate, and nucleoprotein, which is conjugated nucleic acid. So for the last classification of proteins based on chemical differences, we have here derived proteins. So these are proteins that are basically derived by partial to complete hydrolysis or from the simple or conjugated proteins by the action of acids, alkalis, and enzymes. They include two types of derivative, which are the primary derived proteins and the secondary derived proteins. So first, primary derived proteins are formed by processes causing only slight changes in their protein molecule and its properties. On the other hand, secondary derived proteins are formed by the progressive hydrolytic cleavage of peptide bonds or protein molecules. So that's all for the classifications of proteins based on chemical differences. So as a recap, we have here three different classifications of proteins according to chemical differences. Namely, we have simple proteins, conjugated proteins, and finally derived proteins. For simple proteins, they are composed of only one amino acid. For conjugated proteins, they contain non-protein component in addition to amino acids. For derived proteins, they are formed from simple or conjugated proteins through chemical or enzymatic modifications. Okay, so if you have made it this far in this video, I commend you all for that. Now, down to our second to the last classifications, we have classifications of proteins according to shapes. So before anything else, here we have 5 different pictures of different proteins. As you have noticed, some of them have a spherical shape and some of them are parallel. This is because proteins can either be classified as either fibrous or globular. So to start off, here we have fibrous proteins. These are proteins that are shaped like a hair or strand. They are structurally proteins of animals and are insoluble in water. Aside from that, they are also generally resistant to denaturation. On the other hand, globular proteins are like globs, in which they are spherical soluble in water, and are sensitive to denaturation. So before moving on to the next classification, let us first have another pit stop. So here we have pictures that are also shown a while ago. The same thing as we did last pit stop, I'll be giving you guys 10 seconds to guess their classification according to shape, or you can pause the video for a longer time. So time's up. Here are the answers. So collagen, myosin, and keratin are all classified as fibrous proteins. On the other hand, hemoglobin and insulin are both classified as globular proteins. So if you're able to guess that right, please give yourself 3 big claps. So on to our last classification of proteins. We have here proteins that can be classified according to their biological importance. They can be under structural protein, contractile protein, enzyme, hormones, transport proteins, protective or defensive proteins, storage proteins, or as toxins. So first we have here structural proteins. So from the word itself, structural, they compose of structural elements. They are responsible for the cell shape and movement to provide support to major structures. Some examples we have here are keratin, which is the primary constituent of skin, hair, and fingernails. We also have collagen, which makes up the tendons and developing bones. Lastly, we also have elastin, which are elastic connective tissues. Next, we have contractile proteins. So from the root word, contract, it controls the relaxation of muscles. For example, actin, which are moving filaments in myofibril. Another one is myosin, which are stationary filaments in myofibril. Next, we have here enzymes. In this classification is a biological catalyst. Example are trypsin, which hydrolyzes peptides, and amylase, which hydrolyzes polysaccharides. Followed by the next classification, we have hormones, which regulate metabolism. However, not all hormones are considered protein in nature. For example, insulin, which regulates or controls carbohydrate metabolism. Another example is growth hormones, which stimulates the growth of. We also have here transport proteins, in which are responsible for moving molecules across a membrane within a cell. Some examples of it are hemoglobin, which transports oxygen in the blood of vertebrates, iron-binding globulin, which transports iron in the blood, serum albumin, which transports fatty acids in the blood, and myoglobin, which transports oxygen in muscle cells. So next up, we have here protective or defensive proteins. So from the word itself, protective or defensive, they have a defense mechanism. So for example, antibodies, they prevent invading viruses and bacteria. Aside from that, fibrinogen is also a precursor of fibrin in blood clotting. We also have thrombin, which is a component of clotting mechanism. So moving on to the next classification, we have storage proteins, which helps in storage. Examples of it are casein, which is a milk protein, ovalbumin, zein, and glandin. Lastly, we have toxins, which are toxic. They act as poisons such as venous and animal bites and stings or toxins. For example, snake venom, ricin, and Clostridium botulinum toxin, which causes bacterial food poisoning. So you might be wondering how the different classifications of proteins is encountered in the medical field. So here is an example, which is a photo of an antibody. Antibodies belong to the protective or defensive protein in the biological importance classifications, and they are encountered in an everyday life of a medical professional. They prevent invading viruses and bacteria. So like when you get vaccines, it activates your immune response fighting for foreign substances. So that ends my discussion about the different classifications of proteins. So let's go back to our lesson outcomes and determine if we have met our goals. So first, were we able to differentiate the classifications of proteins according to chemical differences, activities, shapes, and biological importance? Yes. Second, were we able to match the different proteins to their respective classification? Yes. And third and finally, were we able to relate the classification of proteins to real-life situations of medical technology? Yes. So if you ever have any questions, just message me on my channel. That concludes my discussion and congratulating you for making this far on the video. So do remember that protein classification offers a wide variety of structured framework that allows us to explore and understand the diverse functions and roles of proteins in their biological systems. So by categorizing proteins based on their activities, chemical compositions, shapes, and biological importance, we were able to gain insight as to how they operate within their cells and organisms. So I thank you for joining me in this exploration of protein classification, and I hope that you would continue to stay curious and keep exploring in this fascinating world of biochemistry. So thank you and goodbye.